As we age, our bodies enter a state of low-grade, persistent inflammation, often referred to as inflammaging. This chronic inflammatory environment, fueled by accumulated cellular debris, senescent cells, and immune dysregulation, quietly reshapes the internal landscape in ways that are deeply harmful over time. One of the most insidious effects of this state is its suppression of autophagy, a cellular cleanup mechanism vital for maintaining tissue health. When autophagy falters under the pressure of constant inflammatory signals, damaged organelles, proteins, and even entire cells accumulate. This buildup doesn't just impair function, it creates the biological soil in which tumors can take root and flourish, particularly in the elderly. Under normal conditions, autophagy plays a pivotal role in dampening inflammation. It does so by removing sources of cellular stress that trigger inflammatory pathways, such as misfolded proteins, defective mitochondria that leak reactive oxygen species, RLS, and cytosolic DNA fragments that can activate innate immune sensors. By clearing these danger signals, autophagy acts as a brake on overactive immune responses. But in aged tissues, where autophagy is already diminished by nutrient-sensing defects, mitochondrial decline, and epigenetic drift, this regulation becomes less reliable. The result is a feed-forward loop. Inflammation suppresses autophagy, and impaired autophagy permits the buildup of pro-inflammatory signals, which further aggravate the immune system. This pro-inflammatory milieu does more than erode tissue integrity. It creates a permissive environment for cancer initiation. Chronic inflammation contributes to DNA damage through oxidative stress, disrupts the extracellular matrix, and fosters immune evasion by mutated cells. At the same time, suppressed autophagy removes a critical line of defense against early tumorigenesis, the cell's ability to recognize and remove damaged, potentially precancerous organelles or genomic material. In this light, autophagy is not merely a mechanism of cellular maintenance, but a frontline tumor suppressor pathway that is actively dismantled by the inflammatory load of aging. In elderly individuals, this dual failure of autophagy and inflammatory control has particularly severe consequences. The immune system becomes less efficient at clearing senescent or dysplastic cells, while those same cells release cytokines and growth factors that stimulate nearby tissues in harmful ways. This senescence-associated secretory phenotype, SESP, is rich in IL-6, TNF-A, and other inflammatory mediators that not only amplify local inflammation but can also awaken dormant tumor cells or support the growth of already established ones. The absence of functional autophagy further compounds this effect by allowing senescent cells to persist rather than be recycled and replaced. Therapeutic strategies in aging care must therefore address this dangerous intersection. Interventions that restore autophagy, such as intermittent fasting, caloric restriction mimetics, or autophagy-activating compounds like spermidine or rapamycin analogs, may help reduce inflammatory burden by clearing cellular stressors. Likewise, anti-inflammatory strategies, including low-dose NSAIDs, targeted cytokine inhibitors, or senolytics, which selectively eliminate senescent cells, can unburden the system enough to allow residual autophagy to function more effectively. The most promising approaches may be those that gently enhance both processes in tandem, taming inflammation while reviving autophagy, without tipping the balance toward immune suppression or excessive tissue turnover. However, precision is essential. Suppressing inflammation too aggressively in the elderly can impair infection control and delay wound healing. Similarly, boosting autophagy indiscriminately may allow certain cancer cells, especially in early or dormant stages, to survive longer. The goal must be resilience, not a complete silencing of inflammation, nor unchecked recycling, but a return to the youthful harmony between damage sensing, cleanup, and renewal. Ultimately, the interplay between chronic inflammation, autophagy, and tumor formation reveals a central axis of aging biology. In younger bodies, autophagy and inflammation are counterbalanced. One resolves damage, the other reacts to it. In older individuals, that balance collapses, and the unfiltered presence of both cellular damage and inflammatory drive creates a high-risk zone for cancer development. Targeting this interface offers not only the chance to prevent tumors, but to restore vitality to aging tissues. By reinforcing the body's natural housekeeping and tempering its misguided alarms, we move closer to aging not just more slowly, but more safely.